Hello, everyone. My name is Alyssa, and I will be discussing the age-based labeling of platelets. Here is an overview of my presentation, and let's get started with a bit of background. So platelets are a type of cell in the blood that are characterized by having no nucleus, and they have a strange morphological shape as shown here. Platelets also express a plethora of receptors for other cells, including platelets, immune cells, the endothelium, and even pathogens, making them a versatile cell that can interact with many others. Platelets in a mouse have a 5-day lifespan, and this is specifically relevant to my project because whether they are within the younger or older days of this lifespan affects their behavior. Specifically, young platelets are shown to form a clot more efficiently than old platelets. The two roles of platelets that I will discuss are hemostasis and immunothrombosis. Now, I know these are big words, but we'll break them down very quickly. Hemostasis can be thought of as simply blood clotting. It occurs in response to a vascular injury, and it forms a plug to stop the bleeding. This clot that is formed is primarily composed of platelets. Immunothrombosis, on the other hand, occurs in response to a pathogen, and it's mediated by platelet-neutrophil interactions. Its main goal is to slow a pathogen from spreading in the bloodstream. Now, while both hemostasis and immunothrombosis form a clot in the vasculature, the composition of the clot differs in that hemostasis is primarily platelets and immunothrombosis is platelets and immune cells. So that leads to my hypothesis that old platelets are primarily involved in immunothrombosis and young in hemostasis. And I predict that we can visualize this using age-based labeling. The two aims I've accomplished thus far are the generation of antibody fragments and the labeling of platelet populations. So here are my methods and results. For 8.1, I should start off by describing what is an antibody fragment, or a Fab is what I will refer to it as from now on. An antibody has two arms responsible for cell binding, and each of these is an individual Fab. The third region is called the Fc portion, and it mediates nonspecific binding and cell-to-cell interactions. I don't want the cell-to-cell interactions in my observations, so I enzymatically digest the antibody and keep the two arms that are responsible for cell binding. I accomplish this using a Thermo Fisher Fab generation kit, and I specifically digested anti-CD49B antibody because this is a prevalent marker found on platelets. I then visualized my Fab fragments using an SDS page. And as we can see, the Fab fragments appear near the 37 kilodalton marker, which according to the reference SDS page given by the Thermo Fisher kit, aligns in the approximate area. The next step was to conjugate my Fabs to fluorophores. Fluorophores can be bound to the antibody, and when they are excited by a laser, they emit light, and the specific wavelength allows for the identification of the cell that the antibody bound. That also ties into the labeling of platelet populations, because I labeled my platelets using flow cytometry, so I withdrew blood via cardiac puncture from a mouse. I labeled cell populations using fluorophore conjugated antibodies, and I passed them through an attuned cytometer. This cytometer contains the laser that excites the cells, and it also picks up the information of each cell emitting specific wavelengths. I can then analyze this data using FlowJo analysis software. Here is my platelet population in whole blood. So I gated on the platelet population, and then I confirmed its identity using CD49B positivity. I then tried to label the platelet population using my Fab fragments. In panel A, the platelet population was acquired using a logarithmic scale, so that's why it looks different than the linear scale on the previous slide. But I confirmed the platelet identity using CD41, which is another platelet marker. We have a distinct positive and negative peak, confirming that this population is platelets. But when I tried to identify it using my Fab fragments, I had no binding, indicating an issue with my Fab generation or the conjugation. The future aims of my project are to label platelet age groups in vivo using the Fab fragments and to observe the behavior of platelet age groups in immunothrombosis and hemostasis. This can have clinical relevance for the application of platelet therapies because young and old platelets could specifically be applied to instances of hemostasis and immunothrombosis. I conclude that Fabs can be generated and visualized using an SDS page, and that platelet populations can be labeled and visualized using flow cytometry. A big thank you to Dr. Craig, Jenny, my PI, and all of the members of the Jenny lab. Here are my references, and thank you for listening.